So before I dive into what is actually happening inside these myofibrils, I just want to step back out and review really quickly some of the terminology because it's going to be very important for you to understand the structure of the myofiber, the muscle cell, for the rest of today's lecture. So try to keep in mind that what happens during muscle contraction is you're going to have a motor neuron, and a branch of that motor neuron will come to each individual cell or each individual myofiber. And what's going to happen is that motor neuron, when it's excited and passes down an action potential, so if you kind of remember action potentials, if you don't remember all the details to them, don't worry, because we're going to get into it in the nervous system. But if you can kind of remember, that's how we pass the electrical signal down a neuron. So as that action potential moves down that motor neuron, it's going to stimulate this sarcolemma now to pass on its own action potential. And what's going to end up happening is you're going to pass this electrical signal from that motor neuron across, and we're going to talk about how that happens later on. But so when that happens, it's going to excite that sarcolemma, and that electrical signal will be passed across the sarcolemma. Remember, that's a plasma membrane. And then as it washes over that sarcolemma, the electrical signal passes down and washes over this sarcolemma. It's going to move down these T tubules. They're also called transverse tubules. We usually call them T tubules for short, but they are called also transverse tubules. So again, that electrical signal passes down those T tubules, and as it does, it's going to signal the release of calcium from this sarcoplasmic reticulum. And then that calcium is going to play a very important role in the contractile activity that's happening inside these myofibrils. And that's what we're going to talk about today. Okay. So again, that just gives you an idea. And don't forget about these mitochondria. Again, these mitochondria need to pump out the ATP, so they're really, important as well. We're going to talk about the function of ATP today. And then again, you have all of the nuclei here, because it's multinucleate. So again, just kind of a review of what the inside of that muscle cell looks like, and a couple of the terms. Oh, and I forgot to mention the term again, sarcoplasm. So again, the sarcoplasm is just going to be that basically the endoplasmic reticulum, or I'm sorry, not the endoplasmic reticulum. The sarcoplasm is going to be the cytoplasm inside that muscle cell. So again, just terminology. So sarcoplasm equals cytoplasm of a muscle cell. Sarcoplasmic reticulum equals endoplasmic reticulum of the muscle cell. So again, just terminology to kind of get familiar with. And then that sarcolemma is that plasma membrane. So now what we're going to talk about today is the contractile units inside these myofibrils. And so if we could pull out these myofibrils, what we're going to see is here where we see this banding, okay, there's going to be this regular pattern of filaments in each of these bands that will make up our contractile unit. And so each one of these is kind of the functional contractile unit that we call a sarcomere, okay? So the basic unit of contraction of your skeletal muscles is the sarcomere. And so we're going to really break it down and talk about what happens inside these individual filaments, basically the microfilaments inside the myofibrils. So again, inside these myofibrils, we're going to have these contractile units that we call sarcomeres. And we're going to talk about what creates this light and dark banding today. And you'll see here, they're going to call the dark band the A band and the light band the I band. So however you can kind of remember that, you may have to come up with your own mnemonic device to remember that. But we're going to talk about what makes that dark band dark and why the light band is light. So let's get into the actual sarcomere. So when you look at the sarcomere here is going to be this contractile unit that's bound by these Z lines. So it's going to go from Z line to Z line. So our Z lines are actually going to be these, we'll kind of talk about them later on. They're going to be these proteins that are called alpha actinin, and they're actually going to help hold these thin filaments together. Now, these thin filaments are going to actually be your actin filaments, and these thick filaments are going to be your myosin filaments, and I'll show you why they're going to look different in just a minute when I show you the structure of them. So again, the thin filaments are the actin filaments, and they're going to extend from the Z line or Z disc, same thing, from that Z line in toward the center of the sarcomere. And the center of the sarcomere here is going to be marked by what's called the M line, which I know you don't see here. I'm going to show it to you in, let me show it to you in this picture here. So here is going to be your M line, okay? And so let me go back and we'll go through some more of these structures. So again, your thick filament is going to be in the middle, basically. It's located right in the center of the sarcomere. And so again, this is going to be your myosin filament or your thick filament. And you're going to see that you're going to have some overlap of these thin and thick filaments here. And so we'll talk about that. And then we're going to talk about what defines each of these zones where there is no overlap here. So when we talk about things like, oops, let me go back, sorry, here. When we talk about things like the H zone here is this area where you only have thick filament in the center of the sarcomere, okay? So again, you only have the myosin filaments here. Here, the I band is going to be where you only have the actin filaments or thin filaments. And then the A band is going to run the whole length of that thick filament, okay? Now you can see the A band is going to be darker because the myosin filaments are thicker, and so they're going to give it a darker look. And so I'll show you why these myosin filaments are thicker in just a minute. And then here, the lighter coloration that you see for the I bands in terms of the striations, again, is going to be because you're only seeing the thin filaments, the actin filaments. And then here you can see, again, a little bit of that H zone where it's really just going to be your myosin filaments. And so it's a little bit lighter in coloration than, again, the rest of the A band where you have overlap. And then you're going to see here, right down the middle is going to be this, what we call M line. And we'll talk about a little bit later on down the lecture, actually probably in the next lecture, that there are some proteins that hold this together called myomycin. And so that's what gives this line, this appearance, is the myomycin proteins that are linked together, holding this together. And so again, you can see kind of that what's creating the striations in your muscles is basically you've got the I band here and then the A band and the I band. And so it's that again, kind of interchanging thin filaments with the thick filaments and with the thin filaments that are giving you that striation, that pattern of dark and light. Now with the myosin, with the thick filaments, the myosin heads are going to contain an enzyme called ATPase. And this is really important for the function of those myosin heads. And so ATPase, if you remember, is this enzyme. Let me turn on my pen here. Oops, sorry about that. I got back here. Let me go back one. Okay, so this ATPase here is going to be an enzyme. Remember ASE, that ACE signifies an enzyme. And so ATPase actually will take ATP and it's going to facilitate the breakdown of ATP into, oops, not do that there, ATP into ADP. Oh, goodness. My pen writing is to be desired there. ADP, if you can't read what that says, that's ADP. And then plus a phosphate. Okay. So, and that's going to be important because remember the binding and then the releasing of that phosphate is oftentimes what's going to change or cause conformational changes in your proteins. So keep that in mind as we talk about myosin here and the myosin heads. I'm just going to exit my drawing for a minute here. Okay. So again, those A bands, just as a review, the A bands are, again, the running the full length of the thick filament. And actually, I'm just going to advance. I'll talk about all of these on the actual diagram so you can actually see it here. So again, here, you can see the A bands here are going to run that full length of the thick filament. And then the I bands here are going to be a region where there's only thin filaments. So the A bands will have overlap between the thick and the thin filaments, but the I bands will only have thin filaments. And then the H zone here is going to be this zone that has no thin filaments, only thick filaments. Okay. So again, I band has only thin filaments, H zone has only thick filaments, and then the A band is going to have some overlap. And then you'll have this line right here is called the M line, right down the middle. You can think M middle. It's right down the middle of the sarcomere. And then the Z lines here signify the end of the sarcomere. So you're going to see, you're going to have, let me grab my pen again. You're going to have back-to-back sarcomeres. So here is going to be one sarcomere from Z disc to Z disc. Here is going to be, your next sarcomere from Z disc to Z disc. And then here would be your next sarcomere. So again, each sarcomere is defined by, again, the boundary, the Z disc boundary. And then that is what is going to form these striations. So you can see again, though, that dark A band here, right here is what you're seeing here. That's that A band. Okay. So let me go ahead and advance. So this is kind of a nice, I like this diagram of one sarcomere. And again, it just kind of shows you how this sarcomere is kind of structured. So here again is that Z disc, which is really, again, going to be just these proteins that are called alpha actinin that are really, again, kind of zigzagging together and helping to kind of hold these actin filaments in place. And so you can see, again, it's going to connect, and it's going to connect this sarcomere to the next sarcomere. So again, those Z discs are pretty important. And then you can see here, you're going to have your thin filament, which is your actin filament. And you can see now why structurally it's thin. And then here is going to be your thick filament, which is going to be, again, your myosin filament. So here's actin, here's myosin. And again, here is going to be that M line right down the middle. So think M middle. So again, you want to make sure that you get really familiar with this. You should be able to kind of even draw this out. Like you should be able to really quickly, go through and draw a sarcomere for yourself. So, again, you should be able to just put together, if, I was drawing a sarcomere, you could put together these thin filaments here, okay, put a Z line here. And then here you're going to put some thick myosin filaments in the center there of your sarcomere. And then your thin filaments, again, overlapping. Oops. Let me draw there. There we go. And then again, our Z line signifying the end of the sarcomere. Sorry, excuse my horrible drawing. I'm sure you can do a much better job. And then, make sure that you can, again, name out all of the different, all of the different parts of this sarcomere. So you should be able to state that this here is the A band, okay, that runs the length of the thick filament. And you should be able to say that this is, going to be the I band that runs the length of the thin filaments. Here would be, again, the H zone right down the middle here with the M line running through. So again, that's my drawing is horrible because my drawing device here is not the new updated versions of that, but you can get the picture of kind of what you should be able to do. So because you want to know that well enough that you could draw that out on an exam if you needed to, so that you can understand how this sarcomere works. So now what I'm going to show you is an animation of, oops, sorry, not that one here. I'll show you an animation of what's called the sliding filament theory of how contraction works inside these sarcomeres. And so when these muscle fibers get that electrical signal to contract, you're going to see what's going to happen is the sarcomere is actually going to shorten. So here you see, again, in a fully relaxed sarcomere here, you can see the comparison then to a fully contracted sarcomere. So you can see when this fully contracted sarcomere happens, what's going to happen is these myosin heads are going to creep along these actin filaments, pulling these actin filaments together and shortening that sarcomere. So in the contracted state, your sarcomere will actually be shorter. So again, this is what we call the sliding filament theory. And again, it's this idea that these myosin heads are kind of pulling these thin actin filaments, right, closer together and shortening that sarcomere during contraction. So they call that the sliding filament theory. And you can see why, again, when in this animation here, where they demonstrate that. So again, there's contraction, relaxation, contraction, relaxation. So now we're going to get into the kind of nitty-gritties of how this actually happens. We're going to start to put this process together. So when we talk about these myofibrils, inside there are going to be these three kinds of proteins. We're going to have the contractile proteins, which you probably already know now are the myosin and the actin. And then we're going to have the regulatory proteins, which are going to be able to turn contraction on and off by basically covering this binding site between actin and myosin. And this is troponin and tropomyosin. So troponin and tropomyosin are going to work together to regulate contraction. And I'm going to show you how this all happens. And then we do have some structural proteins that we're going to talk about a little bit later on here that are going to provide some proper alignment. So we'll talk about this tough protein titin that is actually going to help align the actin filaments. And then we'll talk about myomesin. Again, myomesin is what makes up that M line. And then we'll talk about nebulin. It's another filament that helps to align the actin filaments, and then dystrophin. So we'll get into those in a little bit here, but I thought I'd just introduce them. So I would love for you guys to stop here, if you can, stop the lecture here and go pull up my PowerPoint and click on this animation and watch this animation. It's a short, I think, like 3 or 4 minute animation, but it's going to walk you through the whole process. And I think if you can kind of get the big picture process, then it'll help you as we go through each individual step. So if you can, please take the time to do that, and then come back and start up my lecture here. So when we talk about these contractile proteins, right, we're talking about actin and myosin. And what's going to end up happening, what's going to cause, again, the shortening of those sarcomeres is actually these myosin molecules, these myosin heads sticking up, are going to form a cross bridge or kind of a cross link temporarily with actin, pulling these actin filaments towards the center of the sarcomere. And that's, again, what we saw in that sliding filament mechanism. So I'm going to kind of show you, because I can't pull up my video, I created a short video just to show you kind of what this looks like and how I kind of usually in person demonstrate this to most students. So here is my demonstration.
Right, these actin filaments, and we're going to have these myosin heads, and the myosin heads are going to temporarily bind with the actin filaments and kind of pull them along. And so it's going to kind of do this movement. And so we're going to talk about under what conditions can these myosin heads actually kind of form this cross link with actin filaments to be able to move them along. And so this is the main motion, right, for contraction of your muscles is these myosin heads kind of creeping along, if you can picture that. Again, pulling these kind of and shortening these sarcomeres as they pull along this actin filament. So again, I just kind of wanted to give you a visual. Now, the tough part to picture is that we're going to break it down. We're going to talk about just, again, is one myosin head at a time because it gets so complicated. But you have to remember that there's multiple myosin heads attached, again, to this myosin filament. And so they're going to kind of creep along in all of the, on all these kind of, not all these directions, but all at once with all these kind of myosin heads, all these fingers basically sticking out, creeping along that actin filament. So another instructor gave this example, and I think it's a really good example, like if you've ever put on pantyhose, or if you've ever watched someone put on pantyhose, or you put on like a really tight pair of socks, right? You're going to have to like creep that sock up with your fingers. Okay, so you can picture that as being like all of those myosin heads, and hopefully that gives you a little bit of like a visual of how this is actually happening.
Okay, so yeah, then that's the tough part. In terms of, visualizing, we all have in mind that we simplify things so that, it's a little bit easier to learn. So we're going to talk about, the myosin and the myosin heads looking like this, but it actually looks like this. Our myosin molecule looks like this with multiple heads coming off, forming multiple cross bridges with the actin molecules. Okay, but we're going to, again, break it down and keep it simple. So now what you can see down here is, if you look down here, here is the actin molecules. And if you look on the actin molecules, they're going to have this site, this very specific site that allows them to bind with that myosin head. So each of these kind of little, you can picture them kind of pearls on a string here. And you've got two of them kind of wrapped around into what we call the actin helix. And so you'll have each of these kind of, if you could picture these pearls, right, is going to have a binding site for a myosin head. Okay. Now, remember that we have these regulatory proteins, the troponin and tropomyosin. And so tropomyosin is actually going to cover, that's going to be the protein that actually covers the binding site for myosin, for the myosin heads. And troponin will be attached to that. Now, what will end up happening is when a signal is given to that cell to contract, what's going to happen is calcium is, I'm sorry, I'm going to come back here. Calcium is actually going to bind to this troponin, okay? And when calcium binds to that troponin, it's going to undergo this conformational change, and it's going to pull the tropomyosin with it. So it's kind of like, you can picture like if you if you've ever been, standing in line with your dog on a leash, and your dog maybe sees a squirrel and runs off in the direction of the squirrel and pulls you with it. That's what's happening with the troponin and the tropomyosin. So that troponin is pulling the tropomyosin with it when it undergoes this conformational change. And that's what allows that binding site to be free then to actually form that cross bridge with that myosin head. So that kind of gives you, I kind of jumped ahead a little bit, but while I had that image, I just wanted to kind of show you that. And then we're going to go through now the step-by-step process on how that happens. So this is just a microscopic image showing you, again, these myosin, the myosin heads coming off of this thick filament, and then it shows you the thin filaments. So it's kind of a cool microscopic image. So again, remember that tropomyosin and troponin are those regulatory proteins. And again, this is kind of what I just went through. Tropomyosin is going to cover the actin binding site, but troponin, okay, is really the one that's doing most of the work, because troponin is the one that, if it binds to calcium, it's, again, going to undergo that conformational change and pull that tropomyosin with it, freeing up that binding site. Okay, so, and this is just a quick diagram kind of showing you that. So here's the troponin tropomyosin complex. It's just kind of a cross section, so it's harder to picture the tropomyosin as this kind of rope wrapping around the actin. But here you can see, again, it's just showing you that when that troponin binds calcium, that it's going to undergo, again, that conformational change, pull that tropomyosin with it, and then now free up that binding site for the myosin head. Okay, so now we're going to actually put this all together in this step-by-step process. And so this is one of those that, if you struggle with remembering this is one that I suggest you write out the step-by-step process or draw it out, whatever you need to do, put it on your mirror. And again, it's something that you can look at every day when you brush your teeth, because this process is really important to understanding the overall process of muscle contraction. Okay, so in a relaxed state, right, the cross bridges here are disengaged, meaning the myosin heads are not able to bind to the actin, to the myosin binding site on the actin filament here, because the troponin tropomyosin complex is blocking it right now, right? But the myosin heads are cocked and ready to go in what we call an energized state. So these myosin heads have actually already bound ATP. And then remember I said that there's ATPase, right, inside those myosin heads. So the ATPase, as soon as that myosin head binds ATP, that ATPase quickly breaks down the ATP into ADP. Remember, ATP is adenosine triphosphate. ADP is adenosine diphosphate, right? Because now we've clipped off a phosphate from it. So again, now that ATP is broken down into ADP and a phosphate, okay? And so again, now that myosin head is basically in this kind of cocked and ready position, okay? And so right now, the sarcoplasmic calcium levels are low, meaning that calcium here's the sarcoplasm, right? The cytoplasm kind of surrounding all of these filaments, those calcium levels are low. Now, if for some reason, again, you get an action potential, okay, down the motor neuron that's innervating that muscle and these individual muscle cells, that action potential will then trigger, again, an action potential that's going to move across the sarcolemma, remember, down the T-tubules, and then it's going to trigger the release of calcium from that sarcoplasmic reticulum. So as that calcium is released from the sarcoplasmic reticulum, now it's free, right? It's released into the sarcoplasm, and now it's free to bind to that troponin tropomyosin complex. When calcium binds again to the troponin, it's going to cause it to undergo this conformational change. And now it's going to pull that tropomyosin off of that myosin binding site on the actin, now exposing that active site, that active binding site. So again, remember now the myosin heads are ready to go, okay? They are cocked and ready to bind. The only thing that was preventing them from binding was the tropomyosin that was blocking that active site. So now as soon as, again, that conformational change occurs and tropomyosin's pulled off, boom, that myosin head can bind to the active site, okay, forming what we call the cross bridge. So that's a cross bridge between, again, the myosin filament and the actin filament, okay? So now once that occurs, okay, then there's a release of energy. Basically, again, that energy was stored up in that myosin head that was cocked and ready to go, right? Because the ATP had already been hydrolyzed. So now, as soon as it binds, that myosin head binds to the active site, boom, it's going to release that phosphate along with the ADP. But the big thing is that phosphate. When it releases that phosphate, remember, that's going to cause the conformational change. And when it causes that conformational change, that myosin head is going to kink forward and ratchet, okay? And that's going to be that ratcheting action that's going to pull those actin, again, filaments together, shortening that sarcomere. And so this is what we call the power stroke of that myosin head. So again, this tells you that ATP, again, breakdown had already cocked that myosin head ready to go. Now it's going to ratchet along as that ADP and that phosphate are released from the myosin head. Okay. So then here, as soon as that happens, now ATP, the myosin head can immediately bind another ATP. And once it binds that ATP, again, it'll undergo this conformational change and allow the detachment of that myosin head from that active site. Okay. So basically, once ATP binds to that myosin head, then the cross bridge detaches between the myosin and the actin. Okay, and it's basically going to start our cycle all over again. Because again, once that myosin head then binds that ATP, then remember the ATPase inside that myosin head is going to immediately hydrolyze that ATP into ADP and a phosphate group, right? And that's going to recock this myosin head, okay? So it's ready to go. Now, if calcium remains high inside the sarcoplasm, right, then we can go ahead and that calcium is going to bind to the troponin again and make sure that that troponin tropomyosin complex stays off of that active site. So the myosin head can go ahead and bind that active site again, doing that whole process all over again and ratcheting along that actin filament. If for some reason, again, we are no longer getting stimulation to that muscle, right? The motor neuron is no longer telling that muscle to contract and those muscle cells inside to contract, then that nervous innervation will stop, the action potential will stop, and the sarcoplasmic calcium levels are going to start to decrease as the sarcoplasmic reticulum takes back up that calcium and stores it. Okay, and so as the calcium levels drop, then the troponin tropomyosin complex is going to again cover that actin active site. So this is just kind of showing you this whole cycle, and we call this the kind of ratcheting of the cross bridges here. Because again, those myosin heads are going to be kind of ratcheting and creeping along that actin filament as they do this ratcheting movement. And pulling again those actin filaments closer together and shortening that sarcomere. And so again, this is just another diagram of the same thing. I put them here a couple of times depending upon which one. This one may be, a little bit easier for you to visualize. You might want to print this again and put it up on your mirror if it's a little bit easier for you to kind of visualize all those steps. And so again, this is what's happening as those myosin heads, again, are binding to the actin filaments and creeping along again, pulling those actin filaments together. And again, so the myosin head, may bind here and then creep and pull it in. And then next time it's going to bind up here, creep and pull it in a little bit farther. And that's what creates this kind of sliding filament theory. Okay, as those, again, myosin heads are creeping along those actin filaments. So hopefully you can kind of picture that. And this actually is a great animation. It was one I used many, many years ago, and it's still, I think, one of the best animations of this myosin head creeping along here on the actin filaments. And the great part is you can see the calcium is in yellow here, and it's actually binding to the troponin, tropomyosin complex. So it's binding to the troponin. And then you can see as it binds to the troponin, it will cause that conformational change, pulling that tropomyosin, which is blue, they're in blue, off of that active site. So it's a really great animation for you to be able to visualize these myosin heads kind of ratcheting along that actin filament. And then I put here, please watch this. This is a great summary animation. Again, it's a little bit more detailed, but it will kind of summarize all of this process that I just went through, and it'll actually put it together with what we're going to talk about next, which will be how we pass on that message from the motor neuron to the actual individual muscle fibers. Now, because we've just gone through this process, I like to talk a little bit about what causes rigor mortis. So this is actual rigor mortis, and this is my dog demonstrating rigor mortis. But what causes this after, again, an organism expires? What causes this kind of temporary contracted state of our muscles? And if you go back here to this whole process, if you remember that for cross-bridge detachment to occur, you have to bind ATP. Now, if you are no longer living, you don't have that ability to produce ATP. So temporarily, again, until some of these proteins start to break down, temporarily your myosin heads are actually stuck on those actin filaments because you're no longer able to produce that ATP that would allow the cross-bridge detachment. So that's what causes rigor. Now, again, eventually these proteins are going to start to break down and decay, and then this cross-bridge will be detached because of that process. And so again, rigor mortis is only temporary, but I thought that was a good place to end this lecture. So we'll end here, and then we'll pick up with the rest of the story on muscle contraction for the next lecture.